Zinc finger nucleases are a form of targetable DNA cleavage reagent. This reagent implies their invention from parts, and in a sense, they are synthetically constructed. For that reason, they are also called programmable nucleases, and they were constructed in the mid-90s. ZFNs are composed of two parts. One is a zinc finger protein, which serves as a DNA binding domain that you can engineer according to your preference. The second part is the FokI Nuclease domain. Intuitively, the use of ZFN is not different from meganucleases. We discussed in the meganuclease video that large recognition sites are rare. The same rarity idea is also used in ZFNs. With ZFN as well, you need to find a location in the genome for the nuclease to cut. If you supply a donor DNA with homology near the cut location, the HDR pathway will take care of the donor insertion. You can use ZFNs in this way for knock-in. Likewise, you can choose to cut the DNA at two spots on the same chromosome, and in the absence of a donor, the NHEJ mechanism can take care of the DNA repair to give you a knockout. This same idea is used in all targeted genome engineering methods that I have discussed previously in this series, so I won't belabor the same point in this video. Instead, I want to focus on the mechanistic understanding of zinc finger nucleases. Let's understand the parts independently first, and let's begin with the zinc finger proteins. These were discovered in the mid-80s in TF3A, which is a transcription factor for RNA polymerase III. It has 9 zinc finger domains. ZIF268 is another zinc finger containing transcription factor, which was heavily used to understand the function of zinc fingers. In the most simple terms, zinc finger proteins are DNA binding proteins. Typically, one zinc finger domain is composed of 2 beta sheets and 1 alpha helix, and they are remarkably compact, only around 30 amino acids in total length. The 2 beta sheets are at the N-terminus, and the alpha helix is at the C-terminus. And in this compact structure is a zinc ion that is coordinated by 2 cysteines on the beta sheets and 2 histidines on the alpha helix. And for this reason, you will often see them called cis2-his2 zinc fingers, or C2H2 fingers, for the reasons that these amino acids coordinate the zinc ions. Also, out of the few different types, C2H2 is the most commonly found zinc finger family. My representation of zinc finger is a bit nuanced. You will often see that zinc fingers are typically drawn by this loop-shaped cartoon, where the bulging portion is the so-called finger of the protein. In my nuanced representation, the loopy finger is this alpha helix fold. Alright, we said that zinc fingers bind DNA. The finger, or rather specific amino acids in the alpha helix at positions -1, +3, and +6 bind DNA. These noted positions are relative to the official start of the alpha helix fold in the zinc finger structure. Each of the noted amino acid residues generally contact one specific nucleotide in the major groove of the DNA. The specificity is determined by the amino acids in those positions. There is a direction to this binding. Notice that the N to C terminus orientation of the protein and the 5 to 3' recognition strand is in a way anti-parallel. Generally, it is 3 bases or a DNA triplet that is recognized by one zinc finger. But there is this +2 amino acid position which for some odd reason in some zinc fingers can sample the 4th base on the opposite strand. In any case, the generalized rule is that zinc fingers bind 2 to 4 bases, but on average only 3 bases. Great, so now let's discuss the second part, the FokI cleavage domain. FokI is a restriction enzyme. In the video on restriction enzyme, I discussed that majority of molecular cloning relies on type 2P enzymes. The FokI is in the remaining minority called the type 2S enzymes, which are commonly used in Golden Gate cloning. Here's a quick reminder. The P-type enzymes recognize a palindromic DNA and cleave the same recognition sequence. This generally means that for P-type enzymes, you cannot physically separate their DNA binding and nuclease domains. The S-types are built differently. First, their recognition site is asymmetric, and it is bound by the N-terminal domain of the enzyme. The cleavage domain is shifted away from the DNA binding domain, and so it cleaves DNA in a very non-specific way relative to the recognition sequence. This type of mechanism implies that the S-type enzymes are bipartite in nature, meaning that you can physically separate the recognition and nuclease domains without affecting their activity. So one important feature of FokI is the bipartite structure. The second is its requirement for dimerization. Once FokI is bound to the DNA, that one molecule, or monomer enzyme, is not enough to stimulate the nuclease activity. You need a second enzyme bound somewhere else on a different piece of DNA to come in with contact with this monomer, essentially forming a dimer. This dimer is formed by the cleavage domains, which have a physical interface that then triggers the nuclease activity. A free-floating enzyme can also form a dimer, but it does not stimulate the nuclease activity. A bit of a side note, you will see that 99% of ZFNs use FokI nuclease domain. And this is largely a historical thing. It is simply that first type 2S nuclease, where activity separation into distinct physical domains were discovered, was FokI. This also meant that a lot of research, IP, patent, and optimization went into this enzyme. It just became so robust that it simply worked, and no one really wanted to switch to something new. Alright, from these parts, the zinc finger nucleases were built. Essentially, the zinc finger proteins and FokI nucleases were fused to get ZFNs. And now let's get a bit more specific. Suppose this is a specific sequence you want to target or edit in the genome. Given the specificity of zinc finger proteins, you know that a specific type of zinc finger will bind to a specific triplet of DNA through -1, +3, and +6 amino acids in the helix. You can increase the on-target specificity by having 2 zinc fingers in a chain, which means that the recognition is now extended to 6 bases. This is still not enough, so you generally target 9 or so bases by having 3 zinc finger proteins in a chain. In this chain, the C-terminus is fused to the FokI nuclease domain. There is usually a linker that separates the nuclease domain. Zinc fingers typically don't have any linkers between them. Since the genome is nuclear, ZFNs also have a nuclear localization signal at the N-terminus. So this zinc finger binding site, where 3 zinc fingers are typically bound, sometimes it's 4, happens to target about 9 specific bases. The FokI nuclease domain is expected to target this 5 to 7 nucleotide long spacer DNA just upstream of the zinc finger binding site. This, however, is not going to work, because if you remember, the FokI nuclease needs to dimerize to stimulate DNA cleavage. To force that dimerization, the other side of the spacer is also targeted with a specific set of zinc finger nuclease. This way, when 2 sides are bound by ZFNs, the nuclease domains forms a dimer, and that stimulates the DNA cleavage activity. In this way, you have another 9 bases that adds to the total specificity of the ZFN system. Just to make this explicit, you have 2 monomeric ZFN chains. One binds the left side, the other the right side. And in the middle, you have the spacer DNA that gets cut. This means for each target location, you have 6 zinc fingers and 2 nuclease domains. Each monomer is composed of 3 zinc fingers and 1 nuclease domain. The nuclease domains are 600 in length, so that's about 1.2 kb in total. Each zinc finger is about 30 amino acids, that's 90 nucleotides, meaning that the entire zinc finger library takes only about 600 base pairs. So the ZFN system is very, very compact. So you're targeting 18 base pairs of DNA with just 2 kb of construct, which is pretty nice. Also, keep in mind that cleavage only occurs when both left and right side bind the DNA. just one side binding the DNA by itself is not sufficient to cut the spacer. And theoretically, we assume, or rather hope, that one zinc finger binds 3 nucleotides, and the binding of each finger is distinct and non-overlapping. We discussed that the specificity of each finger is due to the specific amino acid residues in and around the alpha helix. This means to get a zinc finger to recognize a new DNA triplet, you simply change the 3 amino acids, or actually the first 6 amino acids in the helix out of the 30 amino acids in the zinc finger. And that simple change in residues at these select positions will change the DNA specificity of the zinc finger. As an example, if you have arginine, lysine, glutamine in the -1, +3, and +6 positions, and say that it recognizes TAA triplet, then if you change those 3 amino acids to leucine, glycine, and serine, the zinc finger now will recognize a completely new DNA triplet. Only 3 or so mutations, and the protein specificity is completely changed. This sounds relatively reasonable on paper, but this is where the problem starts. First, zinc finger DNA binding can overlap. Remember this infamous +2 position which samples the 4th subsite? This means zinc finger binding is not totally modular, and one zinc finger binding influences the neighboring zinc finger. If it was this simple, then you will have 64 DNA triplets and you only need 64 zinc fingers, and the problem is solved. But because of this glitch in the matrix, the zinc finger behavior is hard to predict. For this reason, zinc finger engineering is very challenging. It costs a lot of money, time, and expertise. Even after engineering, their efficiency in design can still be suboptimal. It is hard to even have a reference point for their absolute efficiency. On top of this, zinc finger binding is easily influenced by chromatin structure, DNA methylation, and protein occupancy. Zinc fingers also tend to have high non-target binding, and usually in the ZFN space of 6 fingers, a single finger off-target effect can lead to toxicity. This toxicity is usually because of the random non-target DNA binding or cleavage, or simply because the cells cannot take a lot of ZFN expression. Zinc fingers are not just used for nucleases. You can attach activators, repressor, methylases, and all sorts of proteins to these finger domains to get a different type of engineering system. This is the same idea as using dCas9 to do nuclease-independent engineering. In these alternative systems, since you don't have FokI Nuclease dimerization requirement, this means that there's no requirement for left and right side monomers. A single 9-base recognizing zinc finger chain is sufficient for activity. But the problem of zinc fingers being sensitive to chromatin architecture still haunts these alternative approaches. Despite their popularity in the late 90s and 2000s, zinc finger nucleases were basically the dial-up internet of biotechnology. Fortunately, Talens came around for the rescue, which I will discuss in the next video.